Hey, well, good morning, everyone, and welcome to this meeting of the Central California Coast Steelhead Regional Temperature Study. This is the last in a series of meetings, and in this meeting here today, we are excited to present the final study findings. So I will ask before we get going into our meeting here, if you all could follow just the typical Zoom protocol, and please have yourself on mute when you are not speaking, and please have yourself on camera if you're able to do so when you are speaking. So here is our agenda for our time here today. So we're going to start out just with a quick introduction and overview of this study. And then we will get into two presentations. The first being a presentation of the Central California Coast Steelhead Temperature Tolerance Study. And then the second being a presentation of the recommendations developed by this study's technical review panel. And then we will close out our time here with a half-an-hour discussion or a Q&A session with the technical review panel, where folks on the call here can ask the TRP questions or questions of any of the project participants that are here with us today. So quick overview of the regional temperature study. So the goal of this study was to develop an approach for determining regionally appropriate temperature evaluation guidelines for supporting the beneficial uses of Central California Coast steelhead. So this is a population that's listed as threatened by the National Marine Fisheries Service. And this project included many participants. Valley Water, who was a project lead, and Valley Water hired consultants to work on this project with them, Stillwater Sciences and Exponent. The San Francisco Bay Regional Water Quality Control Board, also a main project partner and project lead. And then there was a technical review panel that was convened that I will introduce you all to in just a moment. And then in addition to the San Francisco Bay Regional Board, there were other regulatory agency representatives. So representatives from the North Coast Regional Board, the State Water Resources Control Board, California Department of Fish and Wildlife, NOAA NIMS, and also the EPA. And then public stakeholders. So many of you here with us today. And then finally, SFEI. So SFEI was asked by Valley Water and the San Francisco Bay Regional Board to convene the technical review panel and facilitate their effort within this study. So this is essentially a 2-year study, began in earnest in January of 2021, and is set to be completed next month with the finalization of the technical review panel's recommendation memo. And here is our technical review panel, or TRP for short. So we are excited to have such renowned scientists working with us on this project. And the TRP is Dr. Mike Diaz, a principal at Watercourse Engineering in Davis. Dr. Nan Fungi, professor and chair at the Department of Wildlife, Fish, and Conservation Biology at UC Davis. Dr. Tony Farrell, professor emeritus in the Department of Zoology and Faculty of Land and Food Systems at the University of British Columbia. And Dr. Peter Moyle, who is Distinguished Professor Emeritus in the Department of Wildlife, Fish, and Conservation Biology at UC Davis. And all of these folks are here with us today, and I want to thank them for their contribution. So this regional temperature study had 2 phases. Phase 1 included preliminary analyses, technical analyses, using existing data throughout the Central California Coast Steelhead Range. So data related to stream temperature, fish presence and absence, fish condition, those types of things. And then those data were then used to conduct a temperature tolerance study. And so Stillwater Sciences led that effort. And in the first presentation you will see here today, Matt Drenner from Stillwater Sciences, who was the study lead, will provide an overview of the study findings. So that was phase 1. So phase 2 involved the convening of this TRP. And so the TRP reviewed study work products like the memo developed by Stillwater Sciences for the temperature tolerance study. and then also developed recommendations for additional work that should be done to support the development of regional temperature guidelines for Central California coast steelhead. Okay, and so today I will be presenting the TRP's recommendations to you, and you will then be able to ask questions and have a conversation with the TRP about those recommendations after the fact. But we are really excited to get your feedback on these recommendations here today. So with that, I will pass the proverbial mic to Matt to give his presentation on the temperature tolerance study. So let me stop sharing here and hand it to you, Matt.
Thank you, Scott. Let me share my screen. Okay. Let me know if you can see the screen.
Yep, we got it.
And are you able to see my pointer as well?
Yes.
Okay, great. All right, well, thanks a lot, Scott, and thanks everybody for attending today. So I'm going to be presenting the results of the temperature tolerance study Scott mentioned, and I'll get right into it. I'm going to start off by giving a very brief introduction, but most of us are very aware of all the issues involving temperature and fish. So I'll kind of run through this pretty quickly and then get into the bulk of the study. So we're all aware that temperature is a critical environmental factor for fish, and that's because of its influence on metabolic rate, energy demands, and ultimately growth and survival. Fish also have ranges of temperature they can tolerate and limits to what they can tolerate. And then, as we're all very aware, temperatures are increasing in freshwater environments due to climate change as well as other anthropogenic influences. So it's making the need to understand the relationships between fish and temperature even more critical. And we're all focused on steelhead for these particular studies for various reasons, but steelhead are also a really great study species for evaluating interspecific variability and the effects of temperature on fish. And this is in part and largely due to their extensive range. So they occur from as far north as Alaska all the way down to Southern California and the Baja Peninsula. And across this range, as you can imagine, they're exposed to a lot of variability in temperature, and that provides opportunities for development of interspecific tolerances to temperature. And across this range, the northern populations are more extensively studied compared to the southern populations. And so that can create some kind of regulatory management implications when we're trying to manage fish at the southern extent of their range that experience very different temperatures. So our research objective was to characterize temperature tolerance of Central California coast steelhead using existing data, and keyword being existing data there. And we asked a few key questions. Can we use existing data to identify upper temperature limits? Is there any evidence that there's interspecific variability within the Central California coast DPS itself? And then can we detect temperature influences on growth? So our study area here is the Central California coast DPS, which is shown outlined on that map on the right-hand side. And you can see that there's kind of different shaded regions, different colored regions within this diversity strata itself, or within the DPS. And these are individual diversity strata, and diversity strata are kind of defined by NMFS for management and recovery purposes. And so the diversity strata are areas or regions within the Central California coast DPS that share similar environmental conditions such as temperature, as well as other environmental characteristics. And so that'll become important here in a little bit. But what you can see is that there is variability across the DPS in terms of conditions. You can see there's kind of patterns of the diversity strata. You have diversity strata that are more kind of on the coastal environment, and there's differences as you move interior as well as you go from north to south. And so to collect data, we reached out to all the different various stakeholders, regional fish biologists, to ask about certain types of data that were available. And we were focused on a few key types of data. The first was data from mykiss surveys that are conducted typically using snorkel or electrofishing. They were also interested in getting continuous temperature monitoring data from locations where people might be deploying data loggers to monitor temperature throughout the year for different reasons. And we're also interested in habitat and growth data. But what I'm going to be presenting most on today is information from the mykiss survey data and the continuous temperature monitoring data. What we found was that physical habitat data and growth data were either limited in availability or not consistently available enough to incorporate it into analyses. So focusing on just the survey, mykiss survey and the continuous temperature monitoring data, we received a lot of data from our kind of regional partners and collaborators, which was great, but there was a lot of issues with the data in terms of pairing the data together. So the mykiss surveys aren't necessarily conducted in locations where there are continuous temperature monitors and vice versa. So to process the data, we matched it within GIS and then had to do some temporal matching. And then the temperature data itself underwent a QA/QC process. As you can imagine, temperature data range from very processed data that was provided to kind of raw data. And so we had to make sure we were accounting for and identifying things like temperature anomalies that could be associated with temperature loggers that were potentially out of water and recording air temperature or just being heated by solar radiation in shallow environments. And then ultimately the data was aggregated into those diversity strata that I was talking about a second ago. And they were also aggregated based on shared similar survey methods or, this in terms of a sampling units that were conducted. And the data analysis that we used, the first was using generalized additive models or GAMs, which we used two different response variables based on the data we had, fish density and fish presence absence. Fish density we were interested in because it could help us identify temperatures that may not be critically lethal, but would indicate that temperatures are high enough to influence metabolic rate and then thereby influence competition. So you might see reduced densities at higher temperatures. And then we were also interested in fish presence absence because that would get at more of kind of the upper limits to temperature tolerance across the range. So we related fish density and fish presence absence to temperature, and we also included additional covariates to try to account for variables that we know are influencing these biological variables. So things like stream order, which is essentially accounting for anticipated changes in fish density or presence absence that we would expect based on position within the watershed itself. So we might expect higher densities in certain locations, such as headwater tributaries. The stream order can account for that. And then year, we can account for some of the interannual variability that we would expect. There were additional covariates that we considered including in the analysis, things like, as I mentioned previously, physical habitat, which the data wasn't consistently available to include, or other variables such as flow or the month or seasonality of sampling. Some sort of indication of date of sampling was of interest, but we found that these other variables were confounded with typically temperature within the models. So we were not able to include all of them in the models itself. So ultimately, we looked at stream order, year, and temperature. Then we're also interested in trying to detect kind of influences on condition of fish, getting at growth in the best way that we could. So we were looking at condition factor, which is essentially fish's length relative to its weight. And we had two different types of condition factor that we were looking at. The first was a population-specific condition factor, and this is the condition of a fish relative to others within the population it was sampled from. So very focused on kind of a population level differences or maybe even a regional difference. But that doesn't get at, so that tells us essentially whether a fish is fat or skinny relative to other conspecifics within that population, but it doesn't tell us whether the fish overall is in good condition. And so we also looked at what we're calling standard condition factor, which is also known as relative weight. And this gets at the standard condition factor uses data taken from mykiss across the west coast of the US. And so using standard condition factor, we can tell if a fish is actually considered, quote unquote, in good condition because it provides a threshold level for condition, where below that threshold, fish would be considered not in good condition. Above it, they're considered in good condition. And so I wanted to briefly get into how we determined what was the most appropriate temperature metric to include in our analyses. There's a lot of different candidates, temperature metrics that are commonly applied within the literature. One of the most common is the maximum weekly average temperature, or MWAT, which is a single temperature, and it's either calculated over entire years, an annual statistics, or it can be done within a defined season. The next that we considered is a daily statistic. So I've kind of brought this little plot figure here on the bottom to help visualize some of these. So if each of these boxes represents a calendar day, and we have a fish survey with the green box there centered. So we have a daily statistics, which could be the daily mean or max on the date a fish survey occurred. We also considered the 7-day average of the daily mean or max, or 7DADG or DADM. And the 7-day average of the daily mean max is essentially a rolling average that's calculated over the course of a season. You can use that to ask what proportion of days or what proportion of the time are over some certain temperature threshold. So in this case, if we were applying this to the survey date, it would be centered around the date a fish survey occurred. And so we ultimately arrived at using a very similar metric. It's basically just the 7-day average of the daily mean or max. It's slightly modified, and it just specifies that we're going to use the 7 days leading up to the date a fish survey occurs. And so we thought that this provided slightly better biological rationale because we're not predicting fish survival or movements into the future, although overall, because it's calculated the same and it's within a relatively narrow period of time, we would expect this 7-day average of the daily mean or max, or sorry, the weekly average of the daily mean or max to be very similar to the 7-day average of the daily mean max. And in terms of using the weekly average of the daily mean versus max, this figure that I've just brought up now is showing the regression of those two variables. So the weekly average of the daily mean is on the bottom x-axis, and the weekly average of the daily max is on the y-axis. And what you can see is they're highly correlated, as we might expect. So essentially, if one is significant, we would expect the other to be significant. But ultimately, we chose to look at the weekly average of the daily max because we were most interested in trying to define upper temperature limits. So now I'm going to get into the results, and I'll first go over some basic data summaries for the temperature and mykiss survey data that was collected, and then I'll go through the GAM model results and finally fish condition. So this first table here is showing the data summaries from the fish survey data that we collected or that was provided to us. And I'll just walk through this table very briefly. On the left-hand column, you have the diversity strata or model that were run. So these are the final individual models that were run. So the first one is the North Coastal Diversity Strata, and that includes data from the Lower Russian River and the Middle Russian River. The next one is the Interior Diversity Strata, which is from the Middle Russian River. And then we have a model for the San Francisco Bay, and this was essentially an aggregate of two diversity strata, the Interior San Francisco Bay and the Coastal San Francisco Bay. And we aggregated those data together into this San Francisco Bay model because it allowed us to increase sample sizes, as well as we were finding very similar trends in the results when these were modeled, when those two diversity strata were modeled separately. And then finally, even though the Napa River is considered a part of the Interior San Francisco Bay diversity strata, we modeled that data separately. As you can see, it used snorkel surveys rather than backpack electrofishing, which all the other San Francisco Bay tributaries and watersheds were sampled using backpack electrofishing. And in addition, the Napa River itself was only provided to us for a single year. I think it was 2004, whereas all the other San Francisco Bay data was from a pretty wide range of years. So we modeled that one separately, and I'll actually not be showing any results from the Napa River itself, and that's provided in the report within the appendices. So overall, you can see there's a really high sample size. So we had over nearly 2,000 different samples. And I'll just point out a few things here. If you look at the fish density column, you can see that both the North Coastal and Interior Bay have a really high sample size in terms of fish density. And this is because the data that were provided to us was in essentially the individual habitat unit rather than a reach scale. And then as you move down, you look at the San Francisco Bay and Napa River, these are reduced data sets compared to the North Coastal and Interior, and that was because these data were analyzed on the reach scale. But overall, even though San Francisco Bay and Napa River were lower sample sizes, you can see there are over 100 sampling units, which is really good for this type of analyses. So we had 18 years of data from 1999 through 2021. Another thing to note is tended to see more sites where fish were present versus absence. If we look at the north coastal and interior diversity strata, for example, the percent of units where fish were present was approximately 90% compared to 10% where fish were absent. So a lot more sites where fish were present, but we have a more even distribution when you look at the other diversity strata. And then one final note, fish length and weight data was only available from the San Francisco Bay. So when we start looking at the condition factor results, all the data for that was from the San Francisco Bay diversity strata. So now this plot is just showing a basic summary of the temperature data that we collected, and I'll walk through this briefly. Here on the y-axis, we have the weekly average of the daily maximum temperature. We focus here on the upper left-hand plot. This is showing the weekly average of the daily maximum for the different diversity strata or models that were run. And then the bottom 4 panels here are the individual diversity strata that are broken out individually, and then we're showing the season or month on the x-axis here. So a few things to point out. We had weekly average and daily maximum temperatures ranging from 10 degrees Celsius all the way up to 30 degrees. And you can see there are some differences here if we look at the different types of box plots. So we have 2 different box plots. One is a solid white, and this is all the temperature data that was provided to us, regardless of whether there was a matching fish survey site. And the gray box here is showing all the temperature locations, all the temperature data from locations where we had matching fish and temperature data. And so you can see here, if we look at the San Francisco Bay diversity strata, the gray box shows a narrower range of temperatures compared to the white box. So that's suggesting that locations where fish surveys occurred tend to be not at the extremes of temperatures that we have available throughout the watersheds. Another thing you can see if we look at the bottom 4 panels here is that there is seasonality in temperature as you increase, and temperatures increasing as you move into summer and then decreasing as you move into fall, which is expected. But the peak temperatures aren't always consistent across the different diversity strata. And so now I'm getting into the actual model results for starting with fish density. So these were the generalized additive models. And I'll walk you through this plot as well. So we have essentially 2 different columns here. We have the left-hand column, which is the x-axis is water temperature, weekly average of the daily max. And then the right-hand column is stream order. Each row in these plots represents an individual diversity strata that the models were run for. And then on the y-axis here, we have fish density. And so a few things to point out here. First off, I'm going to focus on the left-hand, the water temperature results. And I'll briefly mention these tick marks here. You see it along the bottom, along the x-axis, represent the temperatures where we had fish observation data available. And so temperature was significant in all of the models in the North Coastal Interior and San Francisco Bay. The lines here that you see, the black lines, are the model predictions based on water temperature, and those were modeled while holding the other covariate stream order and year constant. And the blue bands I'll just briefly mention are the confidence intervals. So one thing you can tell immediately is there's some variability in relationships among the diversity strata. If we look at the North Coastal, we're seeing evidence for a peak around 21 degrees Celsius. In the interior, we're seeing kind of this almost bimodal relationship predicted, where you have a peak getting close to 25 degrees Celsius. And in the San Francisco Bay model, we have kind of evidence for a similar bimodal relationship with a peak somewhere around 22 or so degrees Celsius. But I will note that with the San Francisco Bay, you can see there's extremely wide confidence bands at this end of the distribution. So there's a lot of uncertainty where that peak actually is. And then stream order was also significant for all models, and the relationship was kind of as we would predict, where you're seeing decreasing densities with increasing stream order, which is essentially saying that as you move from more headwater tributary type streams into mainstem rivers, you see a decrease in fish density, which is what we would have predicted. I'm not going to show any plots for year, but they were significant in the North Coastal and Interior, but not for the San Francisco Bay. And overall, these models explained relatively high amount of the variability in the data. So we had R-squareds from 0.25 all the way up to 0.47, which is really good considering these are in situ conditions, very complex environment. So the models were doing a pretty good job of predicting fish density. So now getting into the results for the fish presence absence, this plot is very similar to the one that I was just showing here. We have two columns with water temperature on the left-hand column and stream order, the x-axis on the right-hand column. And once again, each diversity strata is an individual row. And in this case, we have presence absence on the y-axis going from 0, which is the probability of 0 occurrence, to 1 at the top, which is 100% probability of occurrence. And the tick marks in this case are on both the bottom and the top x-axis. So the bottom x-axis tick marks show locations where fish were absent, and the top tick marks show temperatures where fish were observed present. And so a few things to note here. First, temperature was significant again in all the models, except for the interior diversity strata. As you can clearly see, this looks more or less like a straight line for this particular model. Once again, there are differences in relationships across the diversity strata. For the North Coastal, we're seeing this kind of predicted increase in probability of occurrence across the range of temperatures observed. Interior, there's no real difference. And then the San Francisco Bay, not seeing much of a trend, although you're seeing a really high predicted probability of occurrence across all the temperatures that were sampled. And if we look at the right-hand column now, we found that stream order was only significant for the Napa River data, which isn't shown here. So all the North Coastal, Interior, and San Francisco Bay models, stream order was not significant for these. And once again, year was significant, but only for the North Coastal and Interior and not for the San Francisco Bay. And finally, we had R-squares that were more variable in the models predicting fish presence absence, so ranging from 0.08 to 0.46. And overall, this is kind of indicating that the variables that we were testing, temperature, stream order, and year, were not as good as predicting fish presence absence compared to predicting fish density. And so now getting into the fish condition results, as I mentioned earlier, those previous plots are showing relationships between temperature and presence absence, but what it doesn't tell us is if a fish is occurring at high temperatures, is it in good condition? So this is what fish condition is kind of helping us evaluate. And we had two different types of fish condition that we looked at. The top figure here is showing the population specific condition factor, which, as you remember, that's comparing a fish's condition relative to others within its given population. And then on the bottom here, we're showing the standard condition factor, which uses data collected more extensively across the West Coast. And on the bottom panel here, there's a dashed line around 95. And that is essentially the threshold where if a fish is above that threshold, it's considered, quote unquote, in good condition. If it's below that, it's considered not in good condition. And so the models predicting fish condition based on temperature was only significant for the population specific condition factor, so this top panel here. But as you can see, based on this trend line, it doesn't appear to be a really biologically meaningful relationship there. And using standard condition factor, we found that 89% of fish that were included in this analysis were considered, quote unquote, in good condition. And temperature, again, was not significant for that model. So so when a fish was not in good condition, it didn't appear to be related to the temperatures it was experiencing. So just to kind of summarize and recap those results, what we found was that temperature was associated with fish density as well as presence absence. We also found that the other factors we included that had represented habitats, stream order representing habitat, as well as year, which represented interannual variability, were also very important in the models. And there were some models when we did a model selection procedure that actually indicated those variables were better explanatory variables for fish presence absence or fish density. The fish were present and at high densities all the way up to the highest temperatures that we had in the data set, up to weekly average of the daily maximum of 28 degrees Celsius. And based on the fish condition results, we found no evidence that temperatures were influencing fish condition across the range of temperatures that was available for that data set. And I briefly wanted to mention some of the study limitations here. We're collecting data, provided from various sources across a pretty large region here. So there are some limitations to that data. And the first and foremost thing to mention is survey design. So depending on, who collected the data, they may be collecting it for different purposes. So it's not essentially a randomized study design. For example, somebody might be interested in monitoring a known fish population, and so their surveys are concentrated in locations where they know or expect to find fish, which might be why we have so many sites where fish are present compared to absent. There was also overall limited sites with high temperatures. We didn't find evidence for an upper threshold for temperature tolerance, but we only had temperatures up to 28 degrees Celsius. So perhaps if we had some locations, with 30 degrees Celsius, we might see that kind of decrease that we would expect. There's also some limitations based on survey timing itself. And this specifically relates to the fish condition factor. And if I was to bring up those plots again, what you notice is the highest temperatures we had for that analysis was around 20 degrees Celsius. And in terms of permits, to be able to handle and weigh and measure a fish, you have to be able to collect them and actually capture them. And so that requires electrofishing typically. And permits actually for conducting those type of surveys are typically specified that you can't sample above 18 degrees Celsius. And so we don't have high ranges of temperatures available to look at higher temperatures and relate that to fish condition. There could also be other confounding factors that we weren't able to account for. One of the biggest ones would be flow. We would think that potentially as flows decrease over the course of the summer and into fall, you might see increased, for example, fish densities or increased probability of detecting a fish because fish are congregated into smaller amounts of remaining habitat. And so that flow, as I mentioned, was confounded with temperature itself, so we couldn't include both in the models. But we did look for evidence of some sort of seasonal effect. So if flow was creating denser congregations of fish in smaller amounts of habitat, we might expect to see higher densities of fish later in the season. So we did look at that, but found no evidence that that was occurring. As well, there could be other seasonal influences in terms of both when sampling occurs, as well as fish behavior over the course of a season. One of the things we were concerned with was that if surveys were conducted earlier in the season, so May or June, might have higher densities of fish present because of young of the year fish that have just recently hatched and emerged. And we also looked at that in terms of whether there was evidence for higher densities of fish earlier in the season. We found no evidence that that was influencing our results. Just to conclude,
so Matt, real quick, is this your last slide? Because we are running behind time now. So just want to check in to make sure this is your last slide.
Yeah, I can finish off with this one.
Okay, great. Thank you.
Yeah. So there's evidence that Central California coast mykiss can tolerate higher temperatures than northern populations. And what's that suggesting is there's either these fish possess an increased aerobic capacity or there's enough prey available to offset the increased metabolic demands associated with high temperatures. Overall temperature is important as evidence from its significance in all the models, but the relationships are complex as we would expect. And ultimately, the study is the kind of first steps in characterizing the regional temperatures and the regional thermal physiologies of a mykiss. I can leave this one off. There's some recommendations, but the TRP will present some of those as well. But I'll just finish off with this acknowledgement slide. So as you can imagine, there was a lot of people contributed either data or provided insight or comments throughout this process. I won't go through all these by name, but I'll just leave it here. Thanks.
Great. Thank you, Matt. And we are running behind time. So folks can ask Matt questions in the chat. You can also follow up with him over email as well. And there might be time at the end of the meeting for circling back for questions for Matt, but maybe if you want to start in the chat now, that could be a great way to go. So thank you again, Matt. And if you wouldn't mind stop sharing, I can then start to share once again. Okay. Okay, so now on to the second presentation here, which is going to be an overview of the TRP's recommendations. So this all starts with the guiding questions that were prepared for the TRP. So there were 3 guiding questions that were developed to guide the work of the TRP within this regional temperature study. The first is, what key factors must be considered when determining protective temperatures that support Central California coast steelhead cold water habitat migration and spawning beneficial uses throughout the range of the Central California coast steelhead. Okay, and so this includes factors such as life stages, thermal adaptation, acclimation, food availability, predation, competition, disease, habitat conditions. Okay. The second question posed to the TRP, Valley Water, the San Francisco Bay Regional Water Quality Control Board, and other regulatory agencies are seeking to improve the thresholds associated with protective temperatures that support Central California coast steelhead beneficial uses. So what information could improve the protective temperature thresholds? And then third, what new data collection and/or experimental or model data analyses should be considered for providing information that could improve the protective temperature thresholds for Central California coast steelhead, and how should those analyses be prioritized? Okay, so what more should be done, and how do we decide what comes first? So these are the three questions that were posed to the TRP and guided, for the most part, the work that they did. So the TRP also developed a set of 4 guiding principles that highlights the key traits, the key steelhead traits that need to be considered when developing protective temperature guidelines. Okay, so these principles essentially provide the foundation for the TRP's answers to those 3 guiding questions. Okay, so the first principle, fundamentals of fish thermal physiology need to be appreciated and acknowledged so that existing knowledge base for Central California coast steelhead can be placed within this broader context and knowledge gaps can be identified. Okay, so there's a lot that goes along with this, this guiding principle, but the TRP thought it was important to highlight thermal performance curves, and an example of such curves is shown here. So the idea that for any activity like growth, for instance, there is a relationship with temperature where there's an increase and decrease as temperature increases. But the idea being that a subpopulation that's acclimated or adapted to higher temperatures will have a curve that is shifted to the right. Okay, so then that T-opt, or the optimal temperature or temperatures, are shifted to the right if the subpopulation is acclimated or adapted to higher temperatures. And then within that T-opt, the TRP thought it was important to point out that, unlike shown here, there is really no one single optimal temperature. It really is a range of temperatures that should be considered optimal for different aspects of performance for Central California coast steelhead. TRP thinking this needs to be kind of in the front of mind when thinking about protective temperatures. Second principle, the difference between thermal adaptation and thermal acclimation needs to be recognized and considered in setting these temperature guidelines. Okay, and so the TRP pointed out that there are data that suggests that protective temperature ranges must be based on data collected from species of interest using relevant or possibly surrogate populations or stocks, and that extrapolating from other species or populations may provide only generalities. So if you're looking to get protective temperature guidelines for a certain species or population, you need to be looking at fish from that population. And also, a fish's thermal acclimation and adaptation responses are shaped by 2 primary things: their evolutionary history and by local thermal regimes. And that the interplay between these 2 processes define a fish population's ultimate sensitivity to environmental temperatures. So then the third guiding principle put forth by the TRP is acclimation temperatures can alter the resistance to thermal extremes as well as the thermal performance of activities. And a lot here, but the TRP thought it was important to highlight thermal tolerance polygons. Okay, so the idea being that there are upper and lower temperature limits for activities like reproduction and feeding, and that certain activities have a narrower range, reproduction having a narrower maximum and minimum range than feeding. And then as acclimation temperature increases, both critical temperature, the CT max, increases and CT min also increases. Okay, so this change in temperature tolerance with acclimation temperature needs to be recognized when thinking about protective temperatures for steelhead. And then fourth, a basic understanding of optimal temperatures and their limits and the limits for their application as needed. So back to the idea that there's not one single temperature, it really is a range that should be considered here. So it's imperative to have population-specific thermal performance curves, like what I just showed earlier, that span a fish's full thermal range at hand when establishing these biologically relevant temperature guidelines, and that a temperature range should be used as a guide for assessing protective temperatures for steelhead over their full life cycle rather than a single fixed optimal temperature value. Okay, so these are the principles put forth by the TRP that then helped them get to their answers to the guiding questions. So guiding question number 1, again, what key factors must be considered when determining protective temperatures that support steelhead beneficial uses? The TRP thinks that there are 7 main factors that should be considered, 4 that should be considered primary factors, and 3 that should be considered secondary factors. So the primary factors are, first and foremost, gene flow. So the TRP thinks that genetic diversity can greatly affect a steelhead's response to temperature throughout the DPS, so throughout the Central California Coast Steelhead range, and should very much be considered. Next, thermal adaptation and acclimation. So the TRP points out that Central California coast subpopulations can be adapted or acclimated to different temperatures, which then affects key traits like metabolism, swimming performance, aerobic capacity. Third is life stage. So the TRP thinks it's important to understand the impacts of duration of high temperature exposure at each life stage, going from eggs all the way to adults. And then the 4th primary factor being disease. So data shows that high temperatures can increase the virulence of parasites and fungi that are harmful to Central California coast steelhead. And so this needs to be appreciated when determining protective temperatures. So these are the primary factors, and then 3 secondary factors: food availability. There has been shown to be many times over a direct relationship between food abundance and temperature tolerance. And then predation and competition. Studies show that a combination of high temperatures combined with predation competition negatively affects steelhead growth compared to steelhead just being exposed to high temperatures. Okay, so that needs to be considered. And then oxygen. So there are studies that show that there's often a trade-off between thermal relief and exposure to low DO conditions. And a good example of that is cool groundwater-fed summer refuge habitat that can also be oxygen depleted. So fish are getting what they need with respect to the cool temperatures, but there's a trade-off in the sense that they are not getting the oxygen that they need. So then on to guiding question number 2, essentially what information could improve the protective temperature thresholds for Central California coast steelhead. Well, the TRP thinks that the information available that is directly useful for determining steelhead protective temperature thresholds is limited, and more information is needed related to genetics, distribution, ecology, behavior, how thermal adaptation and acclimation, life stage, disease affect these protective temperature thresholds for Central California coast steelhead, how protective temperatures are affected by food abundance, predation, competition, oxygen availability. And then finally, looking towards the future, there needs to be an understanding of the impacts of global warming on water temperatures that steelhead experience, both in the near term and in the longer term. So then on to guiding question number 3, what new data collection and/or experimental or model data analyses need to be considered for developing these protective temperatures, and then how should they be prioritized? So the TRP thinks that there are 4 primary study types that need to be focused on to help develop protective temperatures for Central California coast steelhead. So habitat condition assessments, so looking at physical habitat that support the various life stages of steelhead, things like flow depth, velocity, substrate cover, water quality. Also in situ experiments, so observational data collection in the field or during experiments conducted in natural settings. Laboratory experiments, so those lab experiments conducted in isolated systems that are performed on multiple life stages, so eggs, juveniles, adults. And then finally, mathematical modeling, So efforts to predict the spatial and temporal responses of juvenile and adult steelhead to changing habitat conditions and changing water temperatures. Okay, so these are the 4 primary study types. And so then how should we prioritize these types of studies? Well, the TRP thought it could be useful to look at an impact difficulty matrix. Okay, so the idea being that on the y-axis here, you have the impact of a given study going from low to high, and then the difficulty or cost associated with that study going from low to high. And then the colors indicate a degree of priority. So what the TRP discussed was that studies that have a low degree of difficulty or cost, but low to high impact, those should be high priority. So there's really no downside to doing the low cost ones, the low cost, low impact ones, and then the low cost, high impact ones would be the ones that should be, prioritized the highest. So anything that falls in this green zone, any study would be high priority. And then conversely, studies that have a moderate to high degree of difficulty or cost and a moderate to low degree of impact, those should be considered low priority. And then studies in between would be medium. Okay, so this is what the TRP used to then prioritize 10 studies that they developed that they thought would help get to protective temperatures for Central California coast steelhead. Okay, so these studies include a genetic survey, monitoring and modeling studies, and laboratory studies. And then on the right hand here, you see the prioritization of these studies. So all of them are considered high priority by the TRP, but within this group, they ranked them from high to essentially medium low. And so I thought it could be useful for the remaining time here in my presentation to just walk through the moving parts of these 4 studies that the TRP considered to be the highest priority, starting with the genetic survey. So within a genetic survey, the questions that could be addressed include: what is the genetic structure of the mykiss populations within the Central California coast DPS? And how does this structure compare to that of populations in other parts of California and further north? And are there genetic differences among watersheds within the Central California coast region? And to what extent do wild fish hybridize with hatchery fish? And are mykiss rainbow trout above dams residual steelhead that can play a role in conservation, or are they genetically resident fish? Okay, so lots to address within a genetic survey. And then in terms of a high-level overview, the TRP recommends non-disruptive tissue sampling throughout the DPS, and then to ultimately use information from that sampling to compare genetic structure to wild and hatchery steelhead populations in other parts of California and up in the Pacific Northwest. And then the idea here being that the results from this genetic survey could then inform all of the recommended monitoring, modeling, and laboratory studies that the TRP put forth within their recommendations memo. Okay, so the TRP thinks that this is an extremely important study to be doing first and foremost. Okay, next up is regional monitoring. Okay, and the questions that could be addressed within this type of study: what are the stream temperatures throughout the year across the DPS region, and how do they vary both daily and seasonally? And where, when, and how often, and for how long do temperatures reach critical levels for steelhead? And the TRP recommends developing a program to continuously monitor flow, water temperature, and the meteorological information that drive flow and water temperature year-round. And that stations within this type of study would need to be established in enough locations to characterize both spatial and temporal variability. And now moving on to laboratory studies, critical thermal maxima study. And the questions addressed here: what are the critical thermal maxima for Central California coast steelhead and their predators? And what is the optimal temperature window, okay, that range of optimal temperatures for aerobic scope for Central California coast steelhead? And the TRP recommends designing a study to identify the temperatures that bound the optimal temperature window, that range of T-opt values, and evaluate how rapidly aerobic scope declines between the upper T-opt value and critical thermal maxima. And the resulting performance curve from this study could be compared to other steelhead populations in the state and further north to determine if local adaptations exist. And the TRP recommends that within this study, researchers should also vary the duration of exposure to temperature near CT max in order to learn about the steelhead population's degree of temperature tolerance. And then finally, a growth laboratory study. And the question addressed here, how does temperature affect the growth rate of juvenile steelhead? And the TRP recommends acclimating groups of steelhead to different temperatures, feeding them at various rates and measuring growth as well as aerobic scope. And then the impact of higher temperatures on growth rates, the TRP feels will ultimately give an idea of steelhead survivability. Okay, so these are 4 of the highest ranking of the 10 study ideas that the TRP came up with. Just a quick snapshot, but the memo that the TRP has developed with all this information provides a lot more detail. So then in closing here, just a few concluding thoughts from the TRP. So the TRP thinks that collecting information needed to understand protective temperatures for Central California coast steelhead will be quite an endeavor that will require considerable coordination among studies and among researchers. And that such an effort would be a large-scale program that could take up to 10 years or more. And that is needed to effectively resolve the temperature requirements of these steelhead, as well as to determine what these fish are going to need for long-term persistence. Okay, what they're going to need as temperature continues to rise and they continue to be stressed. And then the TRP thinks that such a program could ultimately improve the management of streams that benefit not only the steelhead population, but other desirable biota as well, both in the near term and also in the long term, again, as climate continues to change. And that finally, such a program developed within this region could be a model for other agencies in other regions to follow to benefit other steelhead populations. Okay, so that is an overview of the TRP's recommendations. As I just mentioned a moment ago, there's a lot more to this, and the TRP did develop a memo with these recommendations and a lot more supporting thoughts. Okay, so that memo will be available soon where you can get more information